Hi, my name is Ellen Thomas, and today I'll be doing a mock tutoring session for a kid named Charlie. So, hi Charlie, how are you today? That's good to hear. I'm good, thank you. Now, on the chat, you mentioned that you'd like to run over mitosis today. So, first of all, can you tell me how much you know about mitosis? Okay, so you pretty much know that it's responsible for some sort of cell division. That's a pretty good start. So I've made you a little slideshow today, so we can run through what mitosis is in a bit more detail. Now, first of all, we'll just go over what we learned in the previous lesson, see if you've got any questions from that one still. So we went over DNA transcription and translation, and we sort of determined that it's the replication of organisms' DNA in order to produce new proteins. So did you have any more questions from that one, or did you have any difficulty with the homework that you'd like to go through? Okay, no difficulties. That's great to hear. Let me know if you do have any more questions regarding that one, and we can quickly run through it. Yeah, because it's quite an important topic to remember, especially if you're keen on studying medicine later on in life. Now, what we're going to be looking at today is the processes and events of mitosis, mitotic cell division in somatic cells. And this is going to look at sort of like what is the purpose of mitosis? And we're going to also look at what are the 6 phases of mitosis. Now, I've just put a little happy diagram here for you to refer back to maybe once we've gone through the topic, because it is pretty detailed, but once we run through everything, it will make a lot more sense to you. Awesome. So I just wanted to start off by running through a couple of definitions in that learning objective. So just so we can understand it better before we start actually doing questions and running through the content. So mitosis is the process by which a cell replicates its chromosomes and segregates them by producing 2 identical nuclei in preparation for cell division. So essentially what, to put it in layman's terms, is when you go and write a sentence, say writing an English essay or something, you write a sentence and you want it on the same line below, you copy and paste it, and you paste it. So you copy it and you paste it onto the line below. The sentences are exactly the same. And now you've got 2 of them. So essentially it's like doubling something or recreating the same thing again. So also wanted to quickly run over what somatic means, because that's a bit of a scientific term. So somatic is essentially a cell type which contains 2 sets of a chromosome, one inherited from each parent, which is terminally differentiated. Now, to put that more simply, these are the mature cells in your body. So, the cells that make up the skin on the back of my head, these cells are somatic cells. They've reached the end of their genetic differentiation, and they're just performing the functions of those cells anymore. They can't produce new cells other than copies of themselves. So they can't go anywhere else. These cells can't turn into a lung cell, for example. So they have served they've reached their end stage, and they're going to basically serve a purpose in that area from now on. So did you have any more questions? Did you have any questions regarding that? No? Okay, that's good. Now, what we'll do is I'm going to screen share my iPad, and we'll run through what mitosis is on the iPad, because it will make a little bit more sense. All right, let's go share screen. Okay, just counting down now. All right, it's just having a little bit of difficulty starting the screen share. Give me 1 second. Alrighty, so now we've got the iPad set up. Sorry about that. There was a little blip there for a second. Now, what we're going to do is we're going to run through the phases of mitosis on the iPad. So what I'm going to do is I'm going to explain each stage, and we're going to draw a little diagram as we go along. Now, before we start, so purpose. The purpose of mitosis over here. The purpose of it is to produce two genetically identical daughter cells from a single parent cell. So essentially, what we're trying to do with mitosis is grow the amount of cells in our body. Therefore, it's important for growth, development, and tissue repair in multicellular organisms. So I'll write that out here for you. So produce two genetically identical daughter for tissue repair. Awesome. So we'll just highlight the key points there. For genetically identical, for growth, development, tissue repair, and two. So two genetically identical daughter cells from one parent cell for growth, development, and tissue repair. Awesome. Now, as you can see over here on the side, I've put sort of a table of what the phases are. Now, it can look a little bit daunting right now, but what I'll do is we'll run through it and we'll draw a diagram. Over here, actually, I'll just do a little thing here where it's literally one parent, and then we're going to two daughters that look exactly the same. Awesome. So, first of all, we start with interphase. Now, interphase is essentially the cell performing its normal functions and just getting a little bit ready for the process of mitosis, for the next stages of mitosis. We have in this phase, we've just got, I'll explain, put that little definition in here for you. Normal cell function. Awesome. In interphase, there are actually some separate little divisions, and this is just to help describe the phases a little bit better. So you've got the G0 phase, where the cell is just doing its normal things, normal function. Then you have the G1 phase, which is essentially where your cell is condensing its organelles. And is also the step between normal cell function and DNA synthesis. S phase is very important, so we need to remember this one because this is where your chromosomes actually duplicate. And as we learned previously with transcription and translation, that's an essential stage to creating new cells in the body. You have to have the DNA to actually be in the cell so that those future cells can go on and reproduce more ones, essentially. So duplicate chromosomes. And then the G2 phase, which is essentially where you just get final protein synthesis of the chromosomes. And centriole replication. Now, what the cell actually looks like during this time is pretty boring. So we've got our cell with our nucleus, and then in here we've got lots of DNA. And then once again, normal function, cell, DNA. This one is pretty similar again. We've got more condensing happening now. Here we've got, our two chromosomes here, and now we've got 4, so you get duplication of the chromosomes. And then in this one, we've still got our 4 little chromosomes in there, And we've also got, oh, sorry, we've also got, oh, why is it doing that? Centrioles replicating. Cool. Now, so just quickly run over that again. Interphase, we've got normal cell function, preparation for mitosis, and duplication of the chromosomes. So when the cell splits, we have an even number of chromosomes in them, because the cell wouldn't be a somatic cell if it didn't have two sets of chromosomes in it. So we're trying to replicate exactly what the parent cell was from the start. All right. Any questions there, Charlie? No? Okay, we'll run through the next phase, which is prophase. So in this phase, we actually get condensation of the DNA inside the nucleus. And then we also get the centrosomes, which are eventually what will pull the chromosomes to opposite ends of the cells, migrating to opposite sides of the cells. So that's divided into early and late. What I'm just going to write in the prophase section is we're going to have condensation of DNA and generation of poles at either end of the cell. Condensation of DNA, generation of poles at either end. Awesome. And then in early phase, that's when the condensation happens of DNA. So that's when it gets compacted. DNA. And then we have centrosomes moving to opposite ends of the cell. All right. I'm going to draw the diagram here once again. Righty. So this one looks pretty similar to above, like that. And we've still got our little centrosomes on the same side. And then in late prophase, what happens is they go to opposite ends, and we start getting the chromosomes lining up in the direction they're eventually going to get pulled. So that when the cell divides, we've got two chromosomes in each cell. Awesome. Now, do you have any more questions about that phase? I will get you to do some questions later to run through these phases, and we'll also make a little bit of an acronym so you can remember it. But essentially, so far, what we've got is we've got interphase. So your cell is going about its normal functions. You get duplication of the chromosomes, so you go from 2 to 4. And essentially, by the end of prophase, the cell is ready to almost split in two. Prophase is when you get condensation of your DNA and chromosomes, so they're more readily lined to be pulled. And you also get your centrosomes, which are going to do the pulling to either end at opposite ends of the cell. Awesome. So now we will go on to our next couple of phases. So let me just highlight these because I want to make sure you remember which ones are the important phases. Now we go on to metaphase, which is we get chromosomes lined up in the middle of the cell, like so. I'll do this little diagram here for you. So you get 1, 2, 3. Oh, I need to draw a little bit of a big one because that wasn't big enough. It's a bit hard in the space that I've given myself to draw these, but we'll try again. Like that. 1, 2, 3, 4. Perfect. So metaphase, we get chromosomes lined up in the center. Your centrosomes are at either end. Now what happens is we get microtubules, which attach to the chromosomes, like so. Now we're fully ready for the cell to split. So you've got your microtubules in place, and microtubules are essential. They essentially project out of the centrosome and help to pull the DNA apart, or the chromosomes apart. So the purpose of metaphase is ensuring proper distribution of genetic material when we go through the next couple of phases, ensuring proper distribution of genetic material. Before anaphase. Awesome. Then we go into anaphase, which is when we actually get microtubules which split the chromatids. Now, if you remember from a couple of our previous lessons, chromosomes are joined in the middle, and on either side, this is one chromatid, this is one chromatid, and they join together to form a chromosome. What our microtubules are going to do is actually split that, so we get chromatids going in either direction. So microtubules pull chromatids apart. Awesome. This is starting to get nice and dramatic, which is good. This is when we're starting to almost see two cells forming. So let's go like this, 2, 3, 4, and 1, 2, 3, 4. Cool. Nice. So we've got DNA actually splitting at this point. Now, our next phase is telophase. So this is when we get a nuclear membrane forming around our newly little separated chromatids. So formation of nucleus in daughter cells. We'll go in daughter cells. Please let me know, Charlie, if you have any questions while I'm going through this. No questions? Okay, good. So we'll go like so again. We'll go like this. But now we've got a little nucleus around there. And then the same over here. But that's still within the same cell. The final phase is when you get division of the cytoplasm, and that's when the cells officially split. So division, it's called cytokinesis, bit of a big word, but essentially it means division of cytoplasm. Cytoplasm, which is the fluid which cellular contents are essentially held in. So we'll go to daughter cells, and then they've got their DNA in them. Awesome. So those are the phases of mitosis. Now, I thought to help you remember the steps, we could come up with a little acronym together. Have you got any ideas of what we could do for this acronym to help? So we've got IPMATC. No, nothing? Okay, well, I was thinking we could make it IP on the mat, Chris. Or we could even do, because your name is Charlie, we could do IP on the mat, Charlie. So IP, and we'll just put a little on in there. Mat. Ooh, Matt, Charlie. Oh, sorry. Charlie. And that's IP on the mat, Charlie. Cool. Which is interphase, prophase, metaphase, anaphase, ooh, telophase, cytokinesis. Cool. So interphase, once again, cell prepares. Prophase, we've got the condensation of, oh, sorry, no. Yep, no, we've got the condensation of chromosomes. And centrosomes to opposite poles. Metaphase, chromosomes line up in middle. Awesome. Anaphase, sister chromatids pulled apart by microtubules. Awesome. Telophase. Nuclear membrane forms. And then cytokinesis, division of cytoplasm. Awesome. Now, we are running out of time for our session today. So I did want to quickly, what I'll do is just run through a couple of questions with you. First of all, we'll just go a little bit over time today. And I want you to write out answers for these ones, and then we can talk through the answers for you. So first of all, what is the purpose of mitosis? Yep, so that's the correct answer. It is the generation of two identical daughter cells from one parent cell. And what is it important for, Charlie? Okay, growth, development, and tissue regeneration. Very good. Next question. What are the 6 phases? Okay, so IP on the mat, good. So what did that stand for? Interphase, prophase, metaphase, anaphase, good. Telophase, yes, and cytokinesis. Now, what happens in those 6 phases? What's happening? Okay, so interphase. You've got, yeah, that's correct. So the cell is preparing to divide. You're getting replication of chromosomes. Good. Prophase. Yes. Okay, so prophase is the distinction of the chromosomes. You get the chromatids lined up and the nuclear envelope begins to break down. Good job. Metaphase. Good, yep. So chromosomes come to the midline and the centrosomes are on opposite sides. Good. So this is making, why is that important? Yep, so proper distribution of genetic material. Awesome. Anaphase. Yep, sister chromatids split, go to either end of the cell by the microtubules. Nice. Telophase, what's happening then? Okay, good. Nuclear envelope around the cells. And then finally, what is happening? Good. So you get division of the cytoplasm. Now, I'm going to stop screen sharing on my iPad. Let me just sort this out. Okay, perfect. Awesome. Now, just because we're running a little bit over time, so we went through the questions, that's good. And I helped you along with those ones. Now, this is a really good link to some more questions about mitosis that I found for you. I think there's about 10 or something. So if you want to practice some of those later, that would be good. Now, what I want you to do before our next lesson is I want you to draw your own flow chart. So of the stages of mitosis and the substages within them. And then I want you to draw a little diagram of what's happening to the cell next to it. And feel free to Google things and look it up and make it pretty so that it's easier for you to read. And then I also want you to do questions of 1 to 5 in your biology textbook, and we will review the answers next session. Once we've reviewed those answers next session, we will be learning about meiosis, which is another type of cell division. But for now, just remember what mitosis is, and we'll go through meiosis next lesson. Now, thank you so much for your time today, Charlie. Did you have any questions for me so that I can make the lesson better next time? Yeah, okay, so you'd like to do a video maybe next lesson? That's awesome. I'll go find some resources for that. Thank you so much for your time, and I hope you have a good rest of your day.